Hi, everybody. It's my pleasure to introduce Beatriz Luna for her 2018 OHBM Keynote. Bea is the Staunton Professor of Psychiatry and Pediatrics and Director of the Laboratory for Neurocognitive Development at the University of Pittsburgh. As many of you know, she's a pioneer in developmental cognitive neuroscience and is well known for her work that's used a combination of functional and structural imaging methods to characterize the role of prefrontal cortex in the development of cognitive control. She's well known as the founder and inaugural president of the Flux Congress, which is the world's premier meeting for developmental cognitive neuroscience. And she's winner of the NSF Presidential Early Career Award and is a fellow of the Association for Psychological Science. As I learned in a jam session in her basement after the first Flux meeting, she's also quite a talented punk rock singer. And so her department chair's nickname for her is Dopamine, which I think probably reflects the fact that she's both intensely motivating and highly pleasurable to interact with. So please join me in welcoming her.
Good afternoon, everyone. Thank you so much for coming to this talk, and thank you, Russ, for that really nice and funny introduction. I did not know what to expect. So this is a real pleasure and an exciting opportunity that I've actually dreamed about in my past years to one day be able to address OHBM. And now we have accumulated such exciting approaches to understanding a very unique part of the lifespan, which is the transition from adolescence to adulthood. Now, this is the way that we've been thinking about development. Up until the period of adolescence, brain maturation is proceeding primarily as an approach of accumulation. The brain is getting bigger. Abilities that weren't there before are emerging. And that's very important, and it has its own mechanistic trajectory. But by adolescence, everything is already there. And brain maturation now becomes a process of specialization. And this is important because this process of specialization will be determining what adult trajectories are going to be. Now, we're interested in adolescence because it's unique in a couple of very impactful ways. On the one hand, we know that normatively, during adolescence, there is an increase in sensation seeking. And this can lead to risk-taking behavior that can increase mortality and undermine trajectories. However, this is something that is found across cultures and across species. Even rodents will show during puberty heightened sensation seeking and heightened exploration. So that's giving us a clue that there's something neurobiologically that's occurring that is within the context of an adaptive aspect of development. And the belief is that this increased sensation seeking is needed in order to gain the information so that one can specialize and fit within the environmental demands. Another critical aspect of this adolescent transition is that there is heightened risk for the most significant psychopathologies, anywhere from schizophrenia to mood disorders, addiction, suicide peaks at this time. And this is why the National Institutes of Mental Health have been supporting our work for more than 20 years, because they know that there's a clue to be found there about what are the mechanisms that are changing the brain that can establish something like mental illness. Now, importantly to recognize and influencing our approaches is the following: that across mental illnesses, cognitive processes and reward motivational processes are always impaired across any type of mental illness. Those two things will always be targeted. These are also two of the systems that has a protracted development through adolescence. So by looking at how these processes are developing, we can start to probe in what I wish to be more mechanistic type of fashion to understand what is involved in the transition from adolescence to adulthood. Now, because of this understanding of cognition and motivation, predominantly in our field, we are interested in trying to understand what is the relative balance between executive function and affect and motivation, which is very unique during adolescence, where we have continuously found, and others as well, that there might be a predominance in the influence of affective processes. And note that I did not call it an imbalance. It is a unique balance because adolescence is not a disease. It is a very important part of the development that is needed for specialization. Now, predominant dual systems models have emerged that try to, figure out what is the relative contribution of prefrontal executive processes depicted here in a solid line versus those motivational processes that are depicted here in a hatched line. And these are really conceptual, and they're really based on what we know from the phenotypic aspects of adolescence and a little bit of what we know about neurobiology. The earlier models very much emphasize that a lack of prefrontal function is core to understanding adolescence. In our model, in this driven dual systems model, we actually propose that adolescence might be characterized by the arrival of adult-level executive function, and that this is critical to understanding the adolescent period, because this arrival of executive abilities is within the context of heightened influences of motivational systems. So let me tell you a little bit of the evidence that has led to us thinking in this manner. So what I'll show you today is how we have used different neuroimaging modalities, really trying to get more and more mechanistically into understanding the neurobiology of the changes that are particular to this process of specialization. So first, let me tell you what we have found out that has to do with prefrontal cortex in the context of cognitive control tasks. First, let me tell you that we have a lot of these studies will build on this amazing longitudinal data set that we have, where we have been scanning individuals. Now this one's a little bit old. Now they're coming back for their 13th year of assessment. And it was done in a 3T Allegra scanner that has never had an upgrade. Pittsburgh is very scanner rich, and this one has just every year they're about to change and they never do. It's been such an important piece of information that the NIH has asked us to share it, and it is now being shared. So go to town if people are interested in this. So first, let me start as a background, telling you about what we have found in the past that inform this model. We've looked at inhibitory control, we've looked at working memory, and we've done this by using eye movement tasks. Why eye movement tasks? Because these tasks are used at the single cell level with non-human primates. So we know about its neurochemistry, we know about its neurophysiology, and its neuroanatomy, which really supports our ability to interpret the type of data that we obtain. So basically, people have seen it, the anti-saccade task, we say do not look at a light, and adolescents will look at the light and then correct it. And that's very telling, right? And that's what we're trying to find out. With the working memory, we say look at a light, come back a fixation, retain that information for a delay period, and then command your eyes based on that information to where the location had been. So these are two very powerful ways to probe the integrity and the maturation of the neural systems that support executive function. So first of all, we see that behaviorally, there are dramatic improvements that occur from childhood to adolescence, and also from adolescence to adulthood. And we look at the brain to see what could be accounting for these behavioral changes. One thing that is not accounting for these changes is the engagement of prefrontal cortex. I'm showing you there, both for the inhibitory control task and for the working memory task. Remember, these are longitudinal studies. That what we find is that there are great changes that are occurring from childhood to adolescence, where there's less need to use prefrontal cortex to do these tasks because it's so much more difficult for children to perform these tasks. But by adolescence, prefrontal cortex is being used in the same manner as adults. What can explain the differences that we find in behavior? On the one hand, for inhibitory control, we found a very compelling finding, which is that there was greater recruitment of the dorsal anterior cingulate cortex. This is a region that we know provides performance monitoring, and also is the part of the brain that signals when we need to instantiate cognitive control. And we found, in fact, that this task, recruitment of the dorsal anterior cortex, when we controlled for age, correlated with performance. And it actually mediated the relationship in behavior between age and inhibitory control. Now, with regards to working memory, we found something different. We found, again, prefrontal cortex was not the answer. Instead, we found increasing engagement of visual association cortex during a spatial working memory task. And this actually bodes very well with the models proposed by people like Brad Postal and Mark Desposito, where they have used machine learning and have identified that the actual mnemonic information is not being kept in prefrontal cortex, but it's being kept in sensory type regions that are related to the task at hand. So here I'm telling you a story that it's not prefrontal cortex per se, but perhaps the ability to integrate to more specialized regions that optimize an ability that is already there. We've also explored this using MEG. As you know, MEG has a much better temporal resolution and can start to tell us again about different aspects that have to do with mechanisms. In this task, we looked again at the antisaccade. We compared it to prosacades, to just reactively looking at lights that were presented. And what we found, number one, was that yes, dorsolateral prefrontal cortex showed increased beta power, which is reflecting top-down control. During correct trials where the individual was able to stop making a prepotent saccade. And that was not different between adolescents and adults. However, when we looked at the frontal eye fields, and let me just tell you that these were the two regions that differentiated between antisaccades and prosaccades, so very intimately involved in the ability to inhibit a response. We found that FEF was characterized by increased alpha power. And alpha power is recognized as a frequency that reflects actual attenuation of neuronal activity. And this was greater in adults compared to adolescents. We then proceeded, because this is highly, we have the temporal resolution, to apply something like Granger causality and look at cross-frequency coupling. And here we found something that's very telling. I told you the prefrontal cortex is already there, but its ability to engage the frontal eye fields in an effective manner to attenuate, that the activity that will generate that reflexive saccade is getting better with adulthood. So again, the same theme. The local circuitry of prefrontal cortex appears to be there, but its connectivity with engaging specialized regions are getting better. Now, just to remind you, this is for the working memory task, That what we see predominantly, in most of our colleagues who study here, who study development, see that there are robust changes in mean performance in executive function. But there are other things that are important when looking at development. And those who study development know and sometimes are annoyed by the fact that there is so much intra-subject variability when you're young. And that actually decreases with age. A lot of times people complain about it. Oh, these kids are, they're unreliable. You don't know how they're going to test or whatever. But this is telling in many different ways. Number one, it's saying that at the trial level, there are times when even children show evidence of an adult level response. And this is huge. This is saying that the neural mechanisms that support inability are already there. What's changing is the ability to use this tool in a predictable, sustained, and controlled fashion. So we were really interested in investigating what some people might call noise, and for a while in the lab, we were going on with by saying that noise is not garbage. It's a piece of the clue that we need to understand. So we were lucky enough to have David Montes, who came from a single-cell monkey laboratory, where they were actually characterizing how variability at the neural level, at the neuropopulation level, was associated with variability in behavior. So he came in and he said, how can we apply this sort of approach using fMRI in vivo in humans? And what he did was the following. He considered the whole brain and devised and found what are the whole brain patterns that correspond to each of the cognitive components that take place in this working memory task. So he found the pattern for encoding the information, right, looking at this task and bringing it down, for maintaining information online, and then for being able to retrieve that information and generating a voluntary response guided by the information in working memory. And he found that when he looked at that mean pattern, there were no changes with age. If you're encoding, your brain is going to have this pattern of activity. What he found instead is that then he measured the variability in the expression of these patterns. So sometimes it was expressed like this, sometimes it was expressed less. So there's greater variability in the expression, very reminiscent of what had been found in the single-cell monkey studies. And what he found was that, in fact, this variability in the gain of the expression of these patterns was associated with behavioral variability. More telling was that both of these decreased with age in this related fashion. And this is very compelling because it's again speaking to this theme that I've been proposing about specialization. One interpretation that we've discussed is that you can imagine that the brain is young. For many mechanistic factors, it has the ability to really exercise a lot of different expressions, right? And the more that you do something, the more experience that you get, you start to realize, okay, wait a minute, no, this is what I need to actually, be optimal in this cognitive process. And then critical period plasticity of closing factors can come in, such as myelination, and cement that that's what's going to be the adult mode of operation. So I find this a fascinating and unique sort of a finding. We've also probed what we can find out about resting state. And here is a paper that Scott Merrick, who is now at WashU with Nico Dossenbach, back when he was a graduate student in our lab, did. He went and he looked at resting state, and I know you've probably heard a lot about resting state and even developmentally. And he was interested in looking at the segregation of communities and the integration across communities. And the first thing that he found that was very impactful was that the segregation of the architecture, of the resting state architecture of the brain, was not changing with age, not even from childhood. It was so surprising that he actually went and he used Bayesian statistics to ensure that the lack of age differences was in fact correct. What he did find instead was that there were increases in the integration between these already very nicely structured communities. And in particular, in networks like the singular or opercular network, a cognitive network, in its ability to integrate with ROIs from other networks, including sensory networks. So again, the idea of specialization, of already having foundational aspects and now refining how this available circuitry can get better. Now, this motivated him to go a little bit deeper and try to understand what is the oscillatory signature of these changes in whole brain connectivity. So what he did is that he was informed by resting state networks. He used the Gordon parcellation to define regions of interest. And we did a resting state MEG. And in that resting state MEG, he identified these different regions of interest, and he was particularly interested in how they were speaking to one another by measuring face locking values, which is a way to look at how these different regions are coupled. And he looked at this for every frequency, for every single subject, and then he looked at age differences. And what he found was the following. Right here, I'm showing you in red, adolescents, and blue, adults. Just for visualization, we actually did all the statistics using age as a continuous variable. And he found that in the majority of the frequencies, there were no changes with age, except in the theta frequency. And the theta frequency is very interesting. It's a slow frequency. It can depict long-range connections, but it has also been associated with having to do with midline structures and with the ability to alert the brain when cognitive control needs to be instantiated. Now, what's really compelling about this finding is that what he found with development was a decoupling in the theta range with development. One would think, oh, stronger connectivity like we see in other aspects of the brain with white matter and with fMRI. But instead, he saw decoupling. Not only that, but he saw that this age-related decoupling in the theta frequency was proceeding in a posterior to anterior fashion and going well within the midline, very reminiscent of what we think of what anterior cingulate cortex might be doing. He also looked at the different resting state networks, and he found, again, in this case, that the salience network was the one that was showing the greatest developmental effects in decoupling. So what could this mean? So it turns out that this data set, for these subjects, we also had measures of impulsivity. We used the well-characterized UPPS assessment of impulsivity in order to try to understand how these changes might correlate to the phenotype of adolescence. Of course, we found in this population that impulsivity was decreasing with age, as is already known, and we confirmed it. But then when he went and he looked at PLV values of this decoupling in the theta range, he found that the decoupling of frontal to frontal and even frontal, particularly orbitofrontal cortex regions of interest, and even connecting to temporal regions, the more decoupled these regions were, the less impulsivity there was. And this decoupling was providing a mediation between the changes that we see with age and impulsivity. And one possible interpretation could be that when you're decoupled in resting state at the level of looking at through MEG with theta, it might be an indicator of greater flexibility. And we've seen this with bold, resting state, and so forth, where people have found that greater flexibility is actually associated with greater cognition. And you can imagine that lack of flexibility or commitment through coupling could underlie impulsivity, which is something that has to do with being reactive and you can't help yourself. Lack of flexibility. So I've presented to you a set of studies that speak to how we are informing what we think is going on with regards to prefrontal cortex during this particular stage of development from adolescence to adulthood. And the story is that it is available at adult levels, but it is specializing, and it is stabilizing, and it is getting better to integrate with optimal regions that might be underlying these changes that we see, these improvements in executive control. Now, if you are interested in understanding how MEG is telling us about the bold signal, I encourage you to go to David Montes' poster. All right, now I'm going to switch and talk about the other part of the equation. What do we know when we look at the limbic, motivational, affective, emotional system that's so predominant and plays such an important role in adolescence? Now, for a long time, we've been publishing studies using the anti-saccade task, in where we give the opportunity to win a reward. And lo and behold, surprisingly, for the first time, adolescents can do this task in a way that no other lab had found, neither did us, when you do not give them rewards. How could that be? And what we repeatedly have found is that there's increased activity in the striatum, at the same time that there is a heightened recruitment of the executive regions that you would need to get that task, that trial correct, so that you can receive the reward at an even greater level than adults do. So this is really telling us that there's enhancement, that there's this ability for there to be enhanced sensitivity to reward processing in adolescence. I also encourage you to go and see Leah Somerville's keynote. She has done some really elegant work trying to understand this relationship, in particular the connectivity of prefrontal systems with striatal systems. So we've gone a little step further lately, because as you know, reward doesn't occur in a 6-second trial in real life. You are typically involved in a rewarded state. Look at me, I'm super excited giving a talk to you guys. So we were interested in probing what would be the effects of a rewarded state. So this is work that we did with Deepu Murthy, who is now faculty at Temple University. And what he did was the following. He took one of these rewarded anti-saccade tasks, and he removed the task effects, so that what remained was what corresponded to the sustained activity that reflects the state during the task. And the state is a rewarded state because it's a rewarded task. And then we also had them do resting state scans in the same session. He was particularly interested in understanding developmental changes in the mesolimbic system. So he wanted to look, are there changes that are occurring in the functional connectivity between the ventral tegmental area, where dopamine is produced, and the ventral striatum, the nucleus accumbens? And he had already worked, even before he came to our lab, in a way that he could really very elegantly define the VTA, which can be very tiny. And then he identified where the ventral striatum was. And we looked at resting state, and yes, these two areas do talk to one another, but we do not see any differences in their relationship during rest. However, when we look at this rewarded state, we see that when you're younger, you have a much more heightened and stronger coupling between the VTA and the striatum. And this was very specific to VTA striatum and not to VTA caudate nucleus. So really giving us a clue, again, adding to this understanding that there's something about adolescence that has to do with heightened mesolimbic functioning. So next, we really wanted to get more mechanistic and start to look at dopamine. And this is a lot of work that I've done with Bart Larson. And he couldn't make it here, but he's been at many OHBM conferences. And what I'm showing you here is a graph that he did based on the literature, the animal literature that's predominant rodent models, but there's also non-human primate models, that has really informed us that, number one, dopamine is not a unitary process. As a developmental cognitive neuroscientist, I have faulted in, overgeneralizing what dopamine could be. There are many important aspects of dopamine. There are different receptors, and that could mean something. There is dopamine concentration. There is the number of receptors, and there's how are these receptors used. And all of these could have their own developmental trajectory impacting behavior. So what we did was the following. We used a molecular MR machine, and this is a 3T Siemens scanner that also has a capability for PET imaging. So this is great. We can gather both of these things simultaneously. And we're interested in development, so we're studying 12 to 30-year-old subjects. However, it is difficult, although not impossible, it is difficult to do PET studies in minors, 12 to 17-year-olds. So we did not do that, right? And we did, with the adults, inject them with a radioactive isotope, with the radioactivity equivalent of, for example, a trip from the United States to Singapore. So that's all that there is. We all survived. We looked at this in two ways. So in the past, we did a really lovely study with Bart Larson, where we looked at tissue iron in the brain. Not blood iron, tissue iron. And we did this because there are literatures in the Parkinson's field and the restless leg syndrome field where they're very much invested in looking at brain tissue iron as a pseudo proxy to dopamine processing. Now, tissue iron is predominantly found in the basal ganglia in dopamine-rich areas, and it is in fact involved in its synthesis. So here we have an opportunity to have a non-invasive way to take a peek at what could be occurring with dopamine in relationship to what it's doing in the striatum. So everybody obtained a measure of tissue iron. We decided to use R2 prime, not QSM, which I know other people use. We're very happy with R2 prime. And then for the 18 to 30-year-olds, at the same time, we were also acquiring PET measures. And we were interested in two aspects of the dopamine system. The presynaptic aspect that will inform us regarding dopamine availability by tapping into the dopamine transporters that are found presynaptically. And this is called a DTBZ acquisition or a VMAT acquisition. And then we were also interested in understanding, well, how is this dopamine being used? Let's look at markers postsynaptically. So we looked at the density of D2, D3 receptors postsynaptically, and also how they were being used with task. So the first thing that we found was the following, and you can see that we've partitioned the striatum so we can be really precise and see if there are any, differences that are standing out. And we found that, in fact, tissue iron significantly increases with development all the way to the 30s and so on. And let me tell you that that's not entirely surprising. The aging work, aging studies have shown that iron accumulates, and yes, some of it is due to dopamine, but there might be other reasons why tissue iron is increasing. But in fact, what we found was this R2' measure of tissue iron had a very beautiful and strong association with the presynaptic marker of dopamine. And it kind of makes sense because we know that tissue iron is involved in synthesis, so it would make sense it would be something that would be tied to the presynaptic aspect of dopamine. We saw a much weaker association when we looked at the actual density of D2, D3 receptors. Then when we just zone in on these 18 to 30-year-olds, we found the following in these PET markers. We found, number one, that these measures of DTBZ or VMAT of the presynaptic dopamine availability was not changing from 18 to 30 years of age. And we would like to make the proposal that given the tight coupling with the tissue iron, that we can make the inference that perhaps it was increasing previously and it has reached, and we're looking at a time when this has already become established. Now, when we looked at receptor density, we found decreases of D2, D3 receptor density. And this is very compelling because it is, again, a story about specialization. And what we believe is occurring is that through childhood and adolescence, the amount of dopamine that's going to be available might be rising, and then it becomes stable. And at that point, the brain says, all right, we've got all this dopamine. Now go and specialize in a use it or lose it sort of fashion. What are the receptors that you're actually going to need and use? So this is why we think it's so compelling in speaking about specialization. Then we also looked at PET, and this is the newest, hot off the presses, literally getting figures even as early as this morning, looking at reinforcement learning so we can see dopamine in action. And this was done by Finn Calabro and others in the lab. And we did a reinforcement learning task. We had a map that was devised by a grid, a 3x3 grid, and we would allow subjects to choose 2 locations at a time of each one of these boxes, of which each box had a different reinforcement probability. And we were able to then do the typical reinforcement learning model that takes into consideration the learning aspect of understanding where the probability of certain squares are going to be based on the previous regime and the ability to sort out prediction errors. And the first thing that we did was to ensure that model fit was not changing with age. So now we have a really nice, sweet situation where we can look at this model. One very compelling finding that we found, and I know that this is compelling because I presented this to PET experts like Anissa Abigadam. One of the things that really blows them away is that we have 80 subjects, which is unfathomed in the PET world. I did not know that. But also that we have this nice, tight relationship with the bold response that is specific to the nucleus accumbens and specific to those subjects who fit the reinforcement learning model, indicated that there was learning that was occurring through the process of doing this task. So that's very compelling evidence of the effects of dopamine during reinforcement learning. Now, when we look at this behaviorally, we found that performance during the task gets better with age, right? So optimal choices keep improving with age. We also asked subjects to do what we call the MAP test. So after the test was done, we saw if they had learned which were the boxes that had the highest reinforcement learning. And there we found that there was also an improvement with age. Now, something hot off the presses that we have found is that there seems to be an indication for greater exploration at the younger ages in adolescence. Meaning that when you had a choice to choose before a sure bet reward or a box that you really hadn't visited enough, younger ones were attracted to go and understand and get information seeking. What is really interesting, and this literally, I got one of these graphs this morning, is the following, that I told you already, that the D2, D3 receptor density is decreasing from 18 to 30 years of age. And that's a process of specialization. However, when we look at how dopamine is being used, right, applying a gamma function, we find that there is an increase in its use, despite the fact that with age it's decreasing. And we believe, early interpretation, that it really might be showing that as you mature, as you get rid of receptors that you don't need and you have the dopamine that you need, that you might be more effective in actually targeting these dopamine receptors and having better reinforcement learning. When we go back and we start to bring it all together by looking at tissue iron and its relationship to this sort of reinforcement learning, what we're finding up to this point, and this is preliminary, is that this tissue iron in the nucleus accumbens is serving as a mediator between age and improvements in the ability to learn and to retain the information from the reinforcement learning task. So in conclusion, reinforcement learning improves with age as exploration decreases, with parallel decreases in dopamine receptor density, but increased effectiveness of receptor binding during task. Now, if you're interested in how this sort of data can inform us about risk for psychopathology by using a machine learning approach and looking at the distribution of these different components of dopamine in the striatum, I encourage you to go and visit Brendan Turvo Clemens' poster. Now, finally, I'm going to tell you just a quick story about how we can bring these two aspects of adolescent brain development together. How are they relating? I've been telling you separately how they're working. So one approach was, again, led by Bart Larson, who, by the way, is now going to go and do a postdoc at UPenn with Ted Satterwhite. And he looked at our white matter information that we've had, and he used the orientation approach that is available, for example, in DSI Studio. And I mention that because we've actually had think tanks in our laboratory to try to understand the differences that we're finding with tensor approaches versus orientation approaches, and how this might be speaking not only to myelination, but to glia, into water, into number of fibers. In any case, here we're going to be talking about the orientation method. And what he did was very clever. He heard about these convergent zones in the basal ganglia. These are zones where white matters come together and they share information. And he was particularly interested in defining the convergence zones where limbic and cognitive regions would be interchanging information, which will then influence action. So he defined, from resting state parcellations, he defined the regions of interest that had to do with cognitive networks and those that had to do with limbic networks, and then he measured the fibers that were coming into this convergence zone. And he asked, does the ratio of limbic to cognitive white matter fibers change with age? And in fact, that is what he found in the predictive fashion, that when you're younger, there seems to be a greater predominance of white matter fibers coming into these convergence zones, purportedly influencing how action will be determined. And by the time that you get older, now the cognitive fibers have more of a predominance. But that wasn't enough. We wanted to know, well, what is driving this effect? And then we go back to what I've been telling you from the beginning. In blue is the cognitive representations. What he found was that the integrity of the white matter fibers that were coming from cognitive networks was not changing with age, and that this effect was predominantly driven by decreases through adolescence into adulthood into the predominance of white matter fibers coming from limbic regions. And this also goes well with another study where we very carefully looked at both functional and structural connections between different parts of the amygdala and different parts of prefrontal cortex. And this was work done by Maria Jausbakowski, who is now faculty in the lab. And she found that there was a dramatic decrease in this connectivity with age. And this was a new finding, and it was counter to what other people had found. So she went and she replicated these findings using a big data repository, the Philadelphia neurodevelopmental cohort, the PNC, and she found the same results. So that we feel very confident that this is what we're seeing, decreases in white matter and in functional connectivity. So in summary, I've shown you what we have been finding using a bunch of different neuroimaging approaches that in conclusion are showing that executive systems are available, although they're specializing, that there's enhanced motivational aspects that might be crucial to help with specialization, and that together, this is how adolescence is providing a manner for adult trajectories to be defined. And this has led to us now, we have this under review. It's a review paper where, led by Bart, where he's identified in the literature molecular mechanisms of critical period plasticity and association cortex in the brain by looking at different neurochemistry from GABA to glutamate and triggers from dopamine, etc. So in our last study that we just started, we're using 7 Tesla, and we want to understand the difference in the ratio between GABA, glutamate, and these iron indices of dopamine, and we're using 7T because this can actually zone in on neurotransmitter function and not just free-flowing neurotransmitter that happens at 3T. And we're going to see how this affects cognition. So thank you to my wonderful lab. Thank you to all the funding agencies and the Staunton Foundation. And if you like what you heard, we are right now in a process of hiring where we have, obviously, as you can see, a lot of projects and we have support. And it would be great to get enthusiastic, cool, fun people to come and join our lab because seriously, it is a very cool lab. You can talk to my students. There is evidence. There is evidence. And finally, as Russ had indicated, I was the founder and the president of the Flux Conference for Developmental Cognitive Neuroscience. Flux is not an acronym. It is really just reminding developmentalists that we're studying a dynamic system, and we have to understand what are the dynamic processes. This year, it is in Berlin, and it is hosted by Max Planck, and it's going to be great. Thank you very much.